Hi, good afternoon. My name's Craig Southern, and I'm a CDO with Cellatoz Therapeutics. And this year, we actually became a clinical stage biotech who are engaged in the delivery of innovative regenerative cell therapies. By way of background, Cellatoz was established in Seoul, in South Korea in 2017 by our CEO, Jason Glynn. And so far, we've been able to raise $32 million US from Korean VCs and other investors. We have just under 60 staff with expertise in cell therapy R&D. And we have a senior leadership with experience in commercializing cell therapies in Asia. In line with the way we work, we built our own in-house GMP facility in 2019. And to date, we've completed 3 preclinical programs. And this year, we're happy to announce that we were able to commence our first clinical trial. So the nature of our company is embedded in the name Cellatoz. We have the ambition to become an A to Z, or perhaps I should say an A to Z, of proprietary cells with applications across multiple therapeutic areas. We're working with a variety of stem cell sources and covering a number of disease indications. But today, I think I'll just have time to tell you about our 2 lead allogeneic formulations, which are neural regeneration promoting cells, NRPC, and these are neuronal Schwann cell-like cells, which we're developing for the indication of peripheral neuropathy. We also have our own proprietary musculoskeletal stem cells, which we're developing for intra-articular injection for osteoarthritis. So looking at our NRPCs, working with one of our long-term collaborators, Professor Sung-Chul Jung at Awa Women's University in Korea, we've been able to take tonsil tissue mesenchymal stem cells and differentiate them using our protected methods into NRPC cells, which bear a number of important Schwann cell-like characteristics, both in terms of their cell surface phenotype and the secretion of a high level of neurotropic growth factors. Importantly, however, NRPC display one of the key features of Schwann cells, which is the ability to myelinate and remyelinate damaged nerves. So over on your right in the green images, we're co-culturing NRPC with mouse DRG neurons. And on the top, we can see our NRPC cells that are stained green for myelin basic protein moving towards a neuron. Over a matter of weeks, the cells will align themselves along the length of the neuron, wrap themselves around, and form an intact myelin sheath. This stimulated our interest in the possibility of NRPC cells being able to treat demyelinating disease in the peripheral nervous system. So right now, our go-to indication is a rare neuropathy known as Charcot-Marie Tooth disease, and particularly type 1A. There's over 100 different gene mutations that can cause this type of neuropathy, but by far the major group of patients, which is CMT1A, carry a duplication of the gene PMP22 on chromosome 17. This gives rise to demyelination of the nerves in the periphery, and as a result, you get a progressive and degenerative loss of sensation and motor function with muscle atrophy in the periphery. And you can see some typical images of patients with the deformity of their hands and feet. And although this disease is rarely fatal, it causes lifelong disability associated with the pain. So as I say, it's a rare disease affecting about 1 in 2,500. There are no approved treatments. And the actual cost of the illness, the burden, is in America alone, is significant. Okay, so we've decided to employ a well-known model of Charcot-Marie Tooth, type 1A, which is a C22 mouse. Now, this mouse presents a very severe model of CTM1A because it has an extra 7 copies of the human PMP22 gene, okay? And the mouse itself is characterized by a really significant weakness in the hind limbs and poor posture, and this is due to demyelination of the medium to large axons in the peripheral nerves. Now, if I can get this video to play, we're going to see the mutant mouse being placed on a rotor rod, and even though it's trying its best, it hasn't got the strength, and after 11 seconds, it will fall off. If, however, we treat the animal with an RPC and we inject them into the thigh muscle, the GC muscle, the animal now has improved performance, and it can stay on the rotor rod for 3 minutes. We've gone from 11 seconds to 3 minutes. That's comparable to the wild type, which will stay on for 5 minutes. We're not going to watch a 5-minute video, but on the lower panels, we can see that the C22 mouse has these medium to large axons that are very much demyelinated. Following an RPC treatment, you start to see the deposition of myelin around the neurons, the axons, sorry, these black rings. And we're getting close to what we see in the wild-type animal. Okay? So what's going on? We believe that NRPC can actually directly remyelinate damaged neurons, but we think the recruitment and the normalization of PMP levels in endogenous Schwann cells is really important. Okay. At the end of the day, what we think is happening is that the NRPC can kickstart the orchestration of a natural regenerative response. And because of this, we believe that NRPC could hold potential for a range of demyelinating disorders, irrespective of the cause of the disease. So, on the basis of that preclinical data, this year we've been able to establish our first phase 1 trial in Korea. It's an open-label study. We're evaluating the safety and the tolerability of these intramuscular injections in CMT1A patients. So the study is being performed at Samsung Medical Center. We're working with patients who have mild to severe disease, and they're getting low, medium, high doses of cells, 6 injections into each limb. So that's 12 injections in total, and we're treating each of the major calf muscles. Okay? We're looking for safety and tolerability, but we're also going to assess neurological function and see if we can see any early signs of efficacy. So far, no issues with safety or immunogenicity, and the patients actually tolerate the injection protocol quite well. Following our Korean study, next year we hope to do a phase 1/2A in Australia. We're going to borrow from the same protocol, but we're going to introduce repeat administration using high doses of cells, increase the number of patients, and include placebo groups. So CMT1A is our go-to indication, but we're also very interested in expanding into other neurological indications, and this includes acute and chronic disease affecting the PNS and also the CNS. Okay? So right now, CMT1A is running in phase 1, but we've recently been able to complete preclinical data in diabetic neuropathy and also peripheral nerve injury, and that's sciatic nerve injury. I should mention that diabetic neuropathy is really important. It accounts for 45% of the global neuropathy market. Okay. Next year, we also hope to look at some interesting applications, including the ability of NRPC to protect the neuromuscular junctions in the extremities in ALS. Okay. We're also interested to see if NRPC can protect and repair damage that's caused by an inflammatory response, such as the attack of Schwann cell membranes by complement in Guillain-Barré syndrome. And if anybody has expertise in these specific areas, we'd be very interested to talk to you. Okay. So I'll quickly move on to our allogeneic musculoskeletal stem cells, which we call MSSC. Okay, so very briefly, we've been able to generate a novel population of musculoskeletal stem cells. Okay, so from using our proprietary technology, from ES or iPSC starting points, we generate MSSC, and they have the ability to differentiate into musculoskeletal cells in vitro, as you'd expect. But also, if we inject them into specific location in animals, they will differentiate in vivo to chondrocytes, osteocyte, or muscle. So in vivo, the differentiation fate of MSSC is dictated by the cues that they receive from the local tissue environment. So we have innovative technology to generate MSSC, and we were able to file our first patents in 2017. We were then very pleased to see that our MSSC have many functional characteristics in common with an endogenous human skeletal stem cell that was identified in a landmark paper by Chan et al. In 2018. However, our MSSC have a unique phenotype. They also represent a very homogeneous population. And so, in essence, what we think we can do is generate a single allogeneic MSSC product. We can do this at scale under GMP, and then the application of these cells can be decided by the surgeon or the physician in the clinic by injecting these cells into specific anatomical locations, for example, injecting them into the cartilage in a damaged knee. We believe that MSSC could find very broad application across a range of globally significant musculoskeletal diseases. And we've been able to carry out some preclinical studies using some standard models. We've looked at a number of osteoarthritis models. We've looked at repair of bone defects and the stimulation of muscle growth in sarcopenia. And on the basis of this data, we decided to go ahead and submit an IND early next year in Korea to study the effect of MSSCs for mild to moderate osteoarthritis, grades 2 to 3. Okay, so for Cellatoz, 23 has been a very busy year for us. 24 is going to be even busier. We aim to take each of our proprietary cell types, at least into phase 1, starting with in Korea. But our focus remains with CMT1A, and we hope that the output from our Korean and our Australian trials will enable us to file an IND in the States for phase 2 towards the end of 2025. Right now, we're looking to raise further funding to support our clinical activities, and our ambition is that by the end of 25, we'll be able to launch on the Korean Stock Exchange. So in summary, Cellatoz would welcome opportunities for financial or strategic partnership to support our ongoing clinical activities. We have a portfolio of regenerative cell therapies that can address rare and global disease markets. We think our cells can be expanded into multiple clinical indications, and we have a strong IP position. So on the screen, I provide our contact details, and we'd be happy to provide any further information. I'll be happy to answer any questions or talk to you after the meeting, and thank you for your attention.